Big Pharma does not want you to understand this technology. Full stop. CRISPR just learned how to selectively shred cancer cells, including the ones the industry has spent decades calling undruggable. And the silence from major pharmaceutical companies is, in my analysis, the loudest tell you will ever hear. Here is the villain. The modern oncology business model appears to be built not on cures, but on treatment cycles. Chemotherapy, immunotherapy cocktails, targeted inhibitors that cost $10,000 a month and that you take until you die or your insurance runs out. A one-time gene edit that destroys the tumor at its source is not a product roadmap. It is a revenue extinction event. And the evidence suggests that the incentive structure across the pharmaceutical industry is fundamentally misaligned with the goal of actually curing cancer. That is not conspiracy. That is economics. Now let me give you the proof that this technology is real. It is here, and the only things standing between it and your oncologist's office appear to be money and willpower. Point 1. Programmable guide RNA targeting. Traditional chemo is a carpet bomb. It kills everything. CRISPR is a sniper. Researchers have engineered guide RNA sequences that bind exclusively to oncogenic mutations, the specific broken DNA that makes a cancer cell a cancer cell. We are talking about targeting KRAS G12D, a mutation present in roughly a quarter of all human cancers that has been considered undruggable for over 40 years. 40 years. The pharmaceutical industry poured billions into small molecule inhibitors for KRAS and failed repeatedly. A CRISPR construct walks in with a 20 nucleotide address and cuts it out. The cancer cell dies. The healthy cell next door does not even notice. Point 2. Delivery vector breakthroughs. The old knock on CRISPR was always delivery. How do you get the payload into the right cells without triggering an immune response or editing the wrong tissue? Lipid nanoparticle technology, the same platform that made mRNA vaccines possible, now appears capable of tissue-specific delivery with dramatically improved efficiency. Recent work shows engineered LNPs reaching tumor sites with off-target edit rates below 1%. That is not theoretical. That is measurable. And the computational models driving this, AI systems trained on massive genomic datasets to predict guide RNA specificity and off-target risk, are getting better every quarter. This is where machine learning and biotech collide in a way that actually matters. Point 3. Multiplexed editing. This is where it gets terrifying for the old guard. CRISPR constructs can now target multiple oncogenes simultaneously. A single treatment could, in principle, knock out 3 or 4 driver mutations at once. Traditional drug development cannot do this. You would need 3 or 4 separate drugs, each with its own billion-dollar clinical trial, each with its own side effect profile that conflicts with the others. CRISPR does it in one payload, one delivery event, one shot. If you have been paying attention and you realize that the institutions you trust to cure diseases may not be structured to actually do it, then you already know why this channel exists. Subscribe, we are not going away. Now here is the counterargument, and it is a fair one. Clinical translation is hard. Off-target edits, even at sub-1% rates, could theoretically cause new mutations. Immune responses to the Cas protein itself remain a concern. Regulatory pathways for gene editing therapeutics are slow and expensive. All true. But here is what dismantles that objection in 2 sentences. Chemotherapy has a known, devastating side effect profile, and we approve it routinely because the alternative is death. Holding CRISPR to a safety standard that existing cancer treatments would never pass appears to be less about patient safety and more about protecting a trillion-dollar treatment market. So here is where we land. The technology to selectively destroy cancer cells, including the ones we were told could never be targeted, exists right now. The science is published. The mechanisms are understood. The AI-driven computational tools to optimize guide RNA design are accelerating faster than any review board can keep up with. The question is not whether CRISPR can cure cancer. The evidence increasingly suggests it can. The question is whether the institutions that profit from cancer being a chronic condition will ever let it. So tell me, Do you believe Big Pharma will voluntarily adopt a technology that could eliminate its most profitable disease category? Or will patients have to drag them into the future kicking and screaming?